This is lesson 5.9 on biotechnology and genetic engineering. We're going to take a look at these two terms. Genetic engineering is the process of manipulating genes, while biotechnology is the process of manipulating organisms or their components, which could be genes, but for the purpose of making useful products. So we look at these fields that are impacted by these two processes: medicine, forensics, and agriculture. And forensics can be tied to industry in general. So you can see that we're using biotechnology and genetic engineering to manufacture biofuels, learn more different about biomaterials, or in forensics, detection of crime. In the healthcare industry or for medicine, it's helped us do a better job at diagnosing diseases, treating diseases, preventing diseases. So we've had more clinical trials and creation of techniques to try to improve the quality. Agrobiology or agriculture is the practice of, we can do farming. We are actually going to also increase yield of crops or animals that provide food. Maybe it's a modification to try to create a new product that doesn't rot. So we're actually increasing our yield in that way as well. So if we take a look at where a lot of this started, is the Human Genome Project was completed in 2003, and that was an international effort to map and sequence the entire human genome. So that was about a 50-year span to figure out the structure of DNA to what is the entire message on DNA. Cloning is a process that is going to create an exact copy. So whether it's gene cloning or reproductive or therapeutic cloning, the idea is that there is an exact copy of either, again, a gene or an organism. There's still work to do, but science, and this is changing all of the time, but this is just an example of how a donor cell is taken from a sheep. It's placed into an empty egg cell where the nucleus has been removed, and then that cell basically is creating a new organism that's identical to the donor nucleus. Therapeutic cloning strategies, this is kind of what is being looked at now, is the idea of cloning stem cells so that we can have cells that can kind of become anything. And the idea of gene therapy is basically cloning a gene and being able to insert a working copy of the gene into cells of that individual. So that was an article that we looked at in this class that we were trying to fix and remedy a Hunter syndrome. Now, selective breeding has been going on for years, but this idea is also a genetic engineering or biotechnology term, because selective breeding is kind of taking the best, or an organism that has an ideal quality, and allowing it to reproduce with another organism that has another quality. So we have the what we call hybrids. This is a mastiff and a bulldog, and they both had different strengths, but the hybrid is a bull mastiff. Another example is if you take these organisms. This is actually a wild mustard plant, and different portions of that plant were actually raised and selected for their quality, and you have all of these different varieties. So sometimes, we call this artificial selection, because this is somebody purposefully selecting what is going to be changed. I'm going to move on to the idea of recombinant DNA. And in this case, we take the DNA of one organism and we combine it with the DNA of another organism. So we tend to say when we take DNA from one into another, we have genetically modified that organism. So just some information: human DNA can be inserted into bacteria, and we can have the bacteria make something for us, and this is how we make human insulin. We can also use bacteria to be engineered to eat oil spills. We can also have plants that have We can have genes that are inserted that are disease resistant or insect resistant, so that would help us with crop yield. We can also insert genes into fruit so that they are hardier, so maybe they don't brown or they don't ripen as fast or they don't freeze. And what we do know is that 70 to 75% of our food in our supermarket is actually genetically modified. So the next page here shows a little funny for you. He's like, you got to have the right genes. So this guy is getting attacked by little insects, but this one isn't. So if we want to create a vitamin-rich tomato with a carrot gene, we're going to take that gene out of the carrot. We're going to put it into a plasmid, which is in this case a bacterium, that we're going to use a medium that's going to transfer that gene into the growing tomato plant, so that now that tomato plant will have that carrot gene. That's a genetically modified organism. Sometimes when we are taking one gene into another, we call it transgenic. So in some cases, transgenic animals, we've taken a human gene, we've placed it into a host goat, and we've then created a transgenic goat so that we can get milk from that transgenic goat. So we're doing this with some different organisms, and you can see that here of why we're doing this. And just taking a look at genetic engineering and forensics, there's times when we collect a small sample of DNA and we need to run multiple tests. So there is a process that's been developed called the polymerase chain reaction, or PCR. And you can think of it as making millions of copies of one DNA. So if you're at a crime scene, find a small sample of DNA, they're going to make many copies of that DNA prior to running any tests or experimentation. This has also allowed scientists to do extensive research. DNA extraction, you can pretty much take DNA from any living thing. You can see it being extracted here from green split peas, tomatoes, peas. And what happens is that in order to analyze it, it needs to be cut into pieces, into gene-sized pieces. So you can imagine there's an enzyme that acts like little scissors that cuts it into smaller segments. And we're looking for genes of interest, and then we can use gel electrophoresis. DNA is a charged molecule, so it is going to actually separate in this gel. So the different sized genes will separate, and the larger ones don't go as far from where you enter them, but the smaller pieces do travel. And as you compare them, you can compare and look for similarities. So in this case, we have DNA bands. They're like genetic fingerprints. We have a victim, we have a suspect 1, suspect 2, and then we have the crime scene. So of course, the victim's DNA is going to be found at the crime scene. Then we can also see if there's anything in common at the crime scene with either suspect 1 or suspect 2. So it's looking like suspect 1 was also at the crime scene. The more bands in common would indicate a closer relationship. Finally, we're going to talk a little bit about gene expression. And we do know that gene expression is impacted by things like the environment. So you can see at different points in DNA expression that the environment can impact what happens. And we're trying to learn more and more about this every day so that we can possibly manipulate this process. So I leave you with thinking about what are some of the potential dangers of biotechnology, but what are some of the possible benefits of biotechnology?